Hello, welcome to Prairie Conservation Action Plan's Prairie's Got the Goods Week webinar. My name is Caitlin Morose and I am the Stewardship Coordinator with Saskatchewan Prairie Conservation Action Plan, or PCAP. Check out other events going on for Prairie's Got the Goods Week. We have a webinar tomorrow at noon about the value of waterfowl management to native prairie conservation. That's by Corey White with the Water Security Agency. And tomorrow at 3 p.m., Dr. Karen Thompson with the University of Alberta will be talking about impacts of industrial disturbance on soil microbial and plant communities in mixed grass prairies of Alberta. Lastly, if you are in the Regina area tonight, check out Trevor Harriet's presentation about grassland matters, 7 p.m. at the Royal Saskatchewan Museum. Check out our website for more information about these events. I would like to take a moment to note that in-kind support for this project has been given by the University of Alberta and the University of Virginia. This project was undertaken with the financial support of the Government of Canada and the Federal Department of Environment and Climate Change Canada. Now a bit about our presenter. Jessamyn Manson received her BSc from the University of British Columbia and her PhD from the University of Toronto. She was an assistant professor at the University of Alberta for 4 years before moving to the University of Virginia, where she currently teaches. Jessamyn's research focuses on the evolutionary ecology of native bees, and she's particularly interested in how the nectar and pollen from flowers affects bumblebee preference and health. Before we begin, I would like to mention that if you have any questions during the presentation, please type it into the questions section on your webinar dashboard. Questions will be answered at the end of the presentation. And now I will turn it over to Jessamyn.
Thanks, Caitlin. I appreciate the introduction. I'm going to put myself in presenter mode and we'll get going here.
Perfect.
All right. Shall we begin?
Absolutely.
Thank you all for joining me this afternoon, or this evening as it is here in Virginia. I'm very happy to be here to chat with you today about work that I've been doing with many collaborators at the University of Alberta, the Royal Saskatchewan Museum, and the University of Calgary. And we'll be focusing most generally today on pollination in the prairies, but more specifically on assessing the diversity of native bees in Alberta's agroecosystems. If I can change slides. There we go. So I thought I'd start today by talking specifically about the value of biodiversity. And what I mean by this is that we all sort of have an inherent understanding of diversity being a beautiful thing, being having aesthetic and intrinsic value, but biodiversity in and of itself actually has concrete and direct benefits to humans. And some of these benefits are already realized, and some of them exist in sort of their potential, their future benefits. One of the ways that we see the value of biodiversity manifested is in the idea of ecosystem services. And so ecosystem services are this key term that keeps getting thrown around. I thought I'd define it before we got further into the talk. Ecosystem services are essentially the functions provided by nature that improve and sustain human well-being. These are things that nature provides for us, things like nutrient cycling, water filtration, and carbon sequestration. And of course, today we're going to talk specifically about pollination as an ecosystem service. So pollination as an ecosystem service sort of has two major roles. First of all, in natural ecosystem, pollination will help maintain biodiversity of flowering plants, and that maintains sort of that key function of flowering plants that has a real trickle-up effect, a real cascading effect to other trophic levels. In more managed ecosystems, of course, pollination is a key ecosystem service for providing crop pollination, crops that include forage crops, food crops, fiber crops, and fuel crops. And so understanding pollination and managing pollination is pretty critical for sort of human health and human benefits. We manage pollination pretty carefully in many of our in many of our agricultural systems. And so you're very familiar, I'm sure, with some forms of managed bees. And bees are really our primary pollinators in most agricultural areas. In particular, the honeybee is one of the most prominent and most efficient pollinators in most agricultural systems and pollinates a wide variety of crops. We do, however, have many other managed bees that are coming online and are playing important roles in terms of providing ecosystem services. These managed bees include bumblebees. You can see here these are bumblebees in a greenhouse. These are an important greenhouse pollinator, particularly of solanaceous crops, so tomatoes and their relatives, as well as a variety of field crops. They're becoming much more important for those crops as well. In addition to these two social bees that produce relatively a large number of workers and provide very efficient pollination services, we also have other solitary managed bees that are becoming important players in providing ecosystem services in our agricultural areas. So on the far left you can see the orchard mason bee, Osmia, and this bee provides pollination services in orchards for fruit trees. And then in the center, bottom center of your screen, you will see the alfalfa leafcutter bee, a very important pollinator of forage crops, particularly in the southern prairies. So managed bees are a very important component of ecosystem services, particularly in our agricultural areas. And so they play a prominent role when we think about pollination. But of course, pollination is more than that. Pollination happens in natural ways as well. And we have a very diverse group of animals that function as pollinators in both natural and agricultural ecosystems. These pollinators range from our familiar bees and some of our more up-and-coming bees like our orchard mason bees and our bumblebees, as well as things like flies and moths and butterflies. And so these native pollinators, many of these pollinators being native, have an important role to play in our agricultural systems in providing those key ecosystem services. And that's what I want to focus on for the rest of this seminar. In particular, I want to highlight the key importance of these native bees. So native bees, first of all, are the primary pollinators in our natural ecosystems. In many cases, they have co-evolved with plants, and they really dominate the landscape in terms of the diversity of flowers that they can visit and how efficiently they can visit them. Because they have co-evolved with many plants, they are critical for plants that require specialized pollination. And so the maintenance of native bees in these populations really is essential for the maintenance of these native plants in these populations. And furthermore, one thing we often don't consider when we think about native bees is the fact that they are fail-safes for honeybees in many managed ecosystems. As I'm sure you've heard about, honeybees have suffered in many cases some significant losses in certain parts of the world. And under these conditions, it is important for us to find ways to preserve and protect our native bees that can also provide, in many cases, equal or better pollination services for our crops. In fact, the data suggests that native bees are already pretty essential for agriculture. This graph, which is published by Garibaldi, data published from Garibaldi et al., will show you what I mean by that. So on our x-axis, we have a number of different key crops. You'll see some very familiar crops in there as well as some unusual crops. So you can take a look. You see coffee and sunflower and strawberry. And what we have here is these are data that show the contribution that wild insects and honeybees make to pollination, be it number of seeds or number of fruits depending on the crop. And these have been standardized so they can be compared across crops. And what you can see is that the contribution made by wild insects, primarily bees, is significant in all crops across the board. In other words, wild bees are consistently making an important contribution to reproduction of many of our crop plants, often significantly more than honeybees on a per bee basis. So that's very good to know. Unfortunately, native bees are not doing very well. And this is, or in some cases, they're not doing very well. And this is something that has been making the news and has been a concern to everyone involved. And so this is data here you can see on a particular species of bumblebee named Bombus affinis. And Bombus affinis used to be one of the most common bumblebees in the eastern North America, both in Canada and the United States. And the data that you can see here, each of these circles on this map represent, the yellow represents data, historical data, and the size of the circle represents the number of individuals at each site. So a large yellow circle means that historically that site would have had a lot of individuals of this bee. You can see, I believe, 3 sites where there's a small amount of orange on these circles. That amount of orange are the number of individuals found in recent samples. We are seeing a significant decline in this species and in many other species in terms of their abundance and also their range. And in fact, Bombus affinis is famous for becoming the first endangered bumblebee species in Canada. Bombus affinis was listed originally by COSEBIC in 2010, and Bombus affinis is in the process of being listed in the United States as we speak. So these declines are real. The data suggests that they are not just in a single species, but rather in a number of species across North America and globally. And there are a number of potential causes for these declines. Some of these causes are likely familiar to you. So one of the major causes of declines of pollinators includes changes in land use. And so essentially the conversion of natural landscape to agricultural landscape can reduce habitat and resources for pollinators. We also find that the conversion of land to monocultural crops can be problematic because it alters the amount of diet diversity that pollinators have. We are seeing increases in pathogens in some cases in native pollinators. Particular pathogens like nosema have become problematic. And although we do not see necessarily novel pathogens becoming a problem, we are seeing that pathogens are becoming more prevalent. And finally, there is some evidence to suggest that, yes, pesticides, particularly neonicotinoids, are leading or contributing to native pollinator declines. So with all of these challenges in mind, it becomes very important for us to understand pollinator communities more carefully, understand who lives where, how many pollinators live where, what habitat and resources are supporting these pollinators, and how we can go about protecting and monitoring and conserving these pollinators. And that's what I want to talk a bit about today. I want to focus on pollinators, particularly bees in the prairies, and some of the work that we've been doing to try and understand these communities a little better. So overall, I want to emphasize from this sort of introductory material that the sustainability of native bee populations can have significant ecological and economic implications. This is both global and specific to the prairies. All right, so let's focus on prairie bees, shall we? The good news about prairie bees is that they are very speciose. We know from previous studies in the prairies that, that there are at least 387 native bees species present in the prairie grasslands. So this is Manitoba, Saskatchewan, and Alberta. This is the highest number of bee species in any ecoregion in Canada. So we have this wonderful opportunity to look at a very diverse community of pollinators. However, it is interesting to note that even though we know a lot about these pollinators, one thing we don't know is a lot about sort of where they live across the entire prairies. So the data we have for these pollinators is somewhat limited in terms of location. We only have so many collections from so many places. The other challenge we have is that a lot of these specimens that we are using to create this collection are historical. Some of these specimens date back to nearly 100 years ago or more. And given all that has happened in the past 100 years in the prairies in terms of urbanization, alteration of habitat and development of agricultural areas, and of course changing climate, these records may not effectively represent what pollinator communities, what wild bee communities look like across the prairies in today's environment. And so with this in mind, several years ago, my colleagues and I started to think a bit about wild bees in the Canadian prairies, specifically in Alberta, and to try and figure out how we could go about assessing them. So, studying native bees in the prairies. This is just a picture I took from the Alberta Biodiversity Monitoring Institute, which is one of the groups that has supported our research from day one. And one of the most distinctive features of this picture is the diversity of landscapes we have here. You can see some highly developed agricultural areas where we've got crops in place. In particular, you can see some canola that will become important shortly. But you can also see some areas that are less developed, what we consider more natural grassland or rangeland. And this is very representative of the agricultural areas we find in the prairies. We see this sort of patchwork of different types of field uses, some of it being highly intensive, highly managed, some of it being less intensive and less managed. So looking at this landscape and thinking about what we know or rather don't know about pollinators, we decided to look at how native bees are distributed across Alberta's agricultural areas. This project that I'll be describing has been done entirely in collaboration with Dr. Cameron Carlisle at the University of Alberta, and as the fieldwork that I'll be showing you and much of the data I'll be showing you was collected by two graduate students, Ashton Sturm and Monica Kohler, who were really the drivers behind much of this project. So I'd like to give them credit for the work that you're about to see. Okay, so we wanted to think about native bee distribution across Alberta's agricultural area, and we were fortunate enough to get support from several different funding agencies who were also interested and invested in this question. And so with two graduate students and multiple funding agencies, we set up a study, a longer-term study, a large-scale study, looking at comparisons in pollinator communities, wild bee communities, between canola and rangelands in Alberta. And this paired site design is really powerful, at least we think it's really powerful, because we have two very different agricultural areas, but both agricultural. On the one hand, we have canola sites. These are intensely managed sites, high disturbance sites, which can have an effect on pollinators' ability to nest, but also sites that for 2 or 3 weeks a year provide a significant amount of floral resources because of the mass bloom of canola flowers. On the other hand, we have our rangeland sites, and our rangeland sites have intermediate levels of disturbance. In fact, their disturbance levels really vary depending on how intensely they are grazed. Rangeland sites tend to have floral resources throughout the flowering season, throughout the growing season, and provide important habitat for pollinators. But they certainly don't provide that same density of flowers that canola sites have. And so we created this study to look at differences in wild bee or native bee communities across a large transect of the province, comparing these two very different agricultural sites and thinking about how those differences could affect our pollinator communities. Okay, So our survey sites were set up, as you can see here in this map of Alberta. In total, we had 12 clusters of sites. These clusters are sort of indicated with these dots. This transect, which runs essentially from Grand Prairie down to the border, runs about 1,000 kilometers. To our knowledge, this is the largest transect that's ever been conducted in a grassland or rangeland or prairie area. And this transect, along this transect, you'll see it's not a perfectly linear transect. It was dictated in some ways by places we could stay, and also, probably more importantly, by availability of both canola and rangeland sites. And importantly, we tried to pair these sites. So these sites are clustered with range and canola sites in each cluster, allowing us to do some important comparisons in terms of regional differences and local differences. These sites were visited twice per season, so twice in July and August in both 2014 and 2015. So that's 4 sort of visit totals. And they were surveyed by 2 teams of students and their assistants. I really hope to be able to report to you how many thousands of miles we put on our trucks today, but I'm afraid I don't have that data. It was significant, let me put it that way. Okay, so how do we survey for pollinators? This is an important component of the study, and what I'm about to tell you is what we consider to be standard methods for collecting data on pollinators, particularly on bees. So we have 2 methods that we survey pollinators at each of our sites, pan trapping and netting. Pan traps are very simple. They're essentially small plastic bowls that we paint 3 colors. We paint them white, well, they're already white, but we keep them white. We paint them yellow and blue, and these are just favorite colors of many wild bees. These traps are then filled with soapy water, which when a bee visits one of these bowls thinking that it is, or traps thinking that it is a flower, they become trapped by the soapy water, and they quickly die in these traps. And we set these traps up along a transect, about 50 meters, multiple transects at each of our sites to collect pollinators. Pan traps are a very important way of collecting smaller pollinators, particularly solitary bees, which are much more difficult to catch in other ways. Larger bees, however, often avoid pan traps, and so we catch them using netting. And the netting procedure is quite simply that we walk transects at our site and we collect bees that land on flowers. And so this is done sort of systematically at all of our sites to make sure that we have a good sense of pollinators of all sizes that might be visiting our sites. And so we conducted this survey, these pan traps and netting. These happen on the same day at each of our sites. So you can imagine a team of students out visiting 3 to 4 sites a day, quickly putting out pan traps first thing in the morning, and then netting for about 30 minutes at each site, multiple people netting, and then quickly driving around to these sites that are kilometers away from each other to collect all of these pan traps. All of the bees that we collect in our study, the ones that are caught in the pan traps and the bees caught in our nets, are stored in ethanol while we are on the road, and then they return to the lab. In the lab, we have to process these samples. As much work as the field work is, and as many kilometers as we put on our trucks, I would argue that processing samples is the most challenging part of this work. And I'm very fortunate to have excellent students doing this part of the study. So when our samples come back to the lab, the first thing that we do is we actually wash them all. So pollinators need to be washed, and in some cases blow dried and fluffed and then pinned in order to be identified. Often after washing but before pinning, or sometimes before washing, and we will sort our bees. You can see some petri dishes where pollinators have been sorted into sort of size classes and similarities to make it easier to identify them. Once these insects have been washed and pinned, they must be identified using taxonomic guides. Some species of bees are very simple to identify, others are very complicated, and the features associated with identifying bees are highly variable. Sometimes it is sort of proportions of heads length to width, number of hairs on certain legs, veination in the wings, etc. And identifying a species of bee can take somewhere in the order of a minute or two if it's an easy species to over an hour, which I want you to keep in mind when you see our sample size. We have a team of 3 or 4 students who have been working on identifying bees taxonomically, and we receive a lot of support in terms of training and advice from colleagues of ours, particularly Dr. Corey Sheffield at the Royal Saskatchewan Museum, who was instrumental in training my students to identify bees using standard taxonomy. Some species of bee are very difficult to identify to species or some groups, and so in some cases we actually have a more general identification method, and that will become important in a moment. So let me start to tell you a little bit about what we've been finding. We've been working on this project since 2014, and we're just starting to have some data on our results and to start thinking about what all of this means. So a quick summary of the collections that we have right now. First of all, we have 68 sites that we sampled in total, and when you add up multiple visits over multiple years, that's 285 total site visits over the course of our study. So that's a pretty impressive number of site visits. In total, we found that we have 19,142 specimens of individual bees per year. These specimens represent 35 different genera of bees and 170 unique taxonomic units. So that is in lieu of species. Many of our bees are identified to species. Some of them are identified to morphospecies, or sort of general, sort of a subgenera stage, which is as carefully as we can identify them at the moment. And you can just see here on the upper right-hand side of the screen, you can see some of our bees in boxes. On the bottom, the bottom white box there shows you bumblebees, and in the top white box are some very small solitary bees. And we've used data from this collection as part of our outreach component of one of our grants. You can see one of my graduate students, Monica Kohler, in the bottom left-hand side of the screen, presenting some of our bees at the Rangeland Research Institute's field day last summer. Oops, pardon me. So let me just go over some of the results that we've been finding in this study in a most general way to give you a sense of what we're learning about bees in the prairies. So here's just a graph showing you bee abundance by genus. So we've got the 35 genera on the bottom of our graph here on the x-axis, and the number of individual specimens are on the y-axis. These bars are colored 2 different ways, blue representing bees found in canola fields and orange representing bees found in rangeland sites. And as you can see, we have about 7 or 8 species that represent the vast majority of the bees that we find. Sorry, 7 or 8 genera that represent the vast majority of bees that we find. If we look specifically at the top 4 genera, I've got pictures of them here on the sort of inset. You can see that we've got Lasioglossum, which are a small solitary bee, which are our most dominant or most prominent group of bees that we see, followed by Andrena and Halictus, or sorry, Andrena and Bombus. Bombus being our bumblebees, and Halictus. So we have 3 solitary bee species and 1 social bee species that are the most dominant species. Now, data that are not included here, this is just for wild bees, so you won't see data on Apis mellifera, on honeybees. But you can see that there's a lot of variety, and you can see that certain species tend to, or sorry, certain genera tend to be more prominent in certain types of landscapes than others. So for example, Lasioglossums seem to be relatively equally distributed in canola and rangeland sites, but Andrena tends to be much more prominent in canola relative to range. And you'll see that as well in some of the other groups like Melisodes further down. Whereas if you look further down, you look at osmia, you can see that osmia are almost exclusively found in range sites. And so we do see some variety in terms of where we find pollinators, where we find different genera of pollinators. That variety extends if we start looking more carefully at the data in terms of different regions of the province and trying to look at some of those patterns. And so what you're seeing here on the right is one of the sort of a subset of our data. This is showing you bees caught in pan traps in 2014. And I'm just using one representative graph to explain some of the patterns that we see in the data that we're looking at. So first of all, and perhaps not surprisingly, native bee abundance and diversity, so diversity measured as species richness and abundance, they vary across Alberta's ecological regions. So specifically, I'm talking about grasslands, parklands, and boreal regions, sort of running from south to north, grassland to parkland to boreal. This is also, we also see variation between years. So I'm showing you 2014, but 2015 does have slightly different, or actually significantly different, distributions in both rangelands and in canola fields. But we do see some trends that I think we can describe. First of all, we tend to see fewer bees as we move north, and this is probably not a surprise. It's colder up north. There may be fewer habitat that are amenable for bees, and so we tend to see higher abundance of bees in southern grasslands and fewer bees up in the boreal. You can see that trend in this graph. Another important factor that I think we really want to emphasize is that bees are not consistently higher or lower in canola sites versus rangeland sites. This is to say that we don't see more bees or more species of bees always in canola versus rangeland, or always in rangeland versus canola. It really depends on other factors. In this particular graph, you can see that in 2 of the 3 regions we see higher number of bees in canola sites, and in the boreal it's the opposite. But if I were to show you data from 2015, you would see that that is not always the case. And so this is important because it tells us that one agro-eco-region is not fundamentally better than another agro-eco-region. They are just different, and that differences in bee communities are likely associated with other factors, not just with a simple management factor, simply are they canola or are they rangeland. So this work, we are in the process of wrapping up some of this work, and fortunately for us, this is just the beginning. I recently started a new collaboration that's been with Dr. Paul Galpern at the University of Calgary. This collaboration is funded by the Alberta Biodiversity Institute, the Alberta Biodiversity Monitoring Institute, and also by the Canola Council of Canada. And this is work that is just getting started now, and we're asking some new questions about pollinators in the prairies. We're looking specifically at pollinators in canola fields, and we're asking how pollination by native bees contributes to canola yield. Something that we have an inkling of in terms of the importance of insect pollination, but not specifically the importance of native bees in Canadian prairies. And we're using a really interesting approach for this question. We're doing some experimental studies like the exposure you can see on the right, but we're also working with growers to collect precision yield data and look at variation in precision yield as it relates to pollinator communities. We're also going to be looking more specifically at how bee communities near canola fields vary over the growing season, specifically considering the fact that canola is an important resource, but only for part of the growing season, and so thinking about how that might affect pollinator populations. And we will also be looking at landscape features and how landscape features can affect native bees in agricultural areas, specifically the distance between agricultural, intensive agricultural areas and native habitat or remnant habitat. So these are some ongoing questions that we will continue to address when it comes to pollination in agricultural areas in the prairies. One thing that I want to point out before we kind of wrap things up, though, is that it's all well and good for us to understand biodiversity of pollinators in the prairies, but of course, it's really not, we don't have a thorough perspective on this if we just monitor these populations for a short period of time. And so in a perfect world, we would conduct long-term monitoring of pollinators, and this would help us to understand relationships between pollinators and biodiversity and to potentially detect any declines in pollinators and to help find ways to curtail those declines. But our current survey methods are kind of limited. First of all, our current survey methods for collecting data on pollinators are pretty costly and time-consuming. After spending multiple summers doing this work, I can most definitely conclude that it is not something that can be done by a single group of people on a sustainable long-term scale. Also, as I pointed out earlier, identifying pollinators is not an easy task. It's very time-consuming, and it requires a very specific expertise, and there are only a few people in the world with this expertise. So it becomes a challenge when you want to continue doing this monitoring on a different scale. Finally, there is some controversy associated with current survey methods. We collect a lot of bees, and if you are interested in bee conservation, one might argue that collecting and sacrificing a lot of bees may go against that mandate to some degree. The data do suggest that there is no long-term effect on pollinator populations through these type of surveys, but often these surveys are done on a short-term scale. So if we went to a long-term monitoring strategy, perhaps we might want to revisit that idea. So what I'd like to propose is that we need to think about how to do long-term monitoring, which is an important component of understanding and maintaining biodiversity, but also think about potentially different approaches to long-term monitoring. So alternative approaches that we could consider include monitoring specific species. So in particular, we could look at economically or ecologically important species, things like bumblebees, which tend to be very abundant and also are very efficient pollinators, particularly of certain crops. So that's one opportunity that might be helpful for considering a different long-term monitoring strategy. We might try to locate an indicator species, and one of my graduate students, Ashton Sturm, is working on whether or not species such as kleptoparasites, which are bees that take advantage of other bees, parasitize other bees in a social way, could function as an indicator species, in which case we would only need to collect data on that species and not on all the other species out there. We could also consider, instead of monitoring pollinators themselves, monitoring threats to pollinators. So we could start surveying a subset of pollinators and looking for things like prevalence of disease or increases in pesticide buildup in their food sources. And these might be indirect ways for us to consider potential changes in pollinator populations at a long-term level. So overall, one of the things that I want to communicate to you is that we need to support native bee populations, and I think that there are a number of approaches to this. First of all, baseline research is really crucial, and the work that we've been doing in the prairies, it's very laborious and it's been very time-consuming, but it's been very, very important because it allows us to understand where we're at. It allows us to understand where the bees are, how many there are, and where they live, and to start actually thinking more strategically about longer-term monitoring strategies and about conservation in general. We need to consider developing some sort of monitoring strategy. This is something that is sort of in constant discussion and is not yet fully crystallized, but I think it's important for it to be on our radar in order to make sure that we can maintain the biodiversity of these species that provide this key ecosystem service. when we are working on land management, it's important to consider how to protect and promote native bees as we develop different land management strategies. And the good news is that in many cases, these strategies need very, very little adjustment in order to provide sustainable and supportable environments for native bees. And finally, I would argue that conservation is going to only be successful if we have combined efforts with agricultural growers, land managers, government agencies, and of course the public. We need everybody to support this approach and this idea of supporting native bee populations in the prairies, in both more native areas and in agroecological areas, in order to actually preserve and protect these important ecosystem service providers. With that, I'd really just like to acknowledge my co-authors on this study. As I mentioned before, Monica Kohler and Ashton Sturm did all of the field work for this study and are in the process of writing this work up at the moment. This work was conceived and entirely in collaboration with Dr. Cam Carlisle, who's speaking later in the week. I strongly recommend you tune in. And Dr. Corey Sheffield and Dr. Paul Galpern have been essential components of doing this work. This truly is a collaborative prairie, cross-prairie collaboration with this study, and we've had a lot of help from different students over the years, and we are provided with support from a number of different funding agencies who have invested in this mandate. And I appreciate your time. I appreciate you learning about our work. Thank you very much.
Thank you, Jessamyn. That was really interesting. If any of our listeners today have any questions, you can type it in on the question section of your webinar dashboard. And there are a couple here right now. So there's one from the very beginning. There seemed to be far less bumblebees in my market garden in 2016 than previous years. Do you know if last year was a bad year for native bees in Saskatchewan? And that was by listener Michelle.
So I can tell you that bumblebee populations go up and down a lot, and they have a lot to do with the winter before. So because bumblebees hibernate over the winter, snowpack is really critical for their success. In addition to snowpack being important, floral resources are important. And so we tend to see low bumblebee years when there's been a low snowpack and when conditions are dry. And in some years it can truly be, they can be very spotty. I'm not 100% sure if last year falls under those conditions, but I would say that you will see a lot of variation, and so it's entirely possible that they will rebound. I wouldn't to date be worried that this is evidence of direct decline, but they are highly variable. So those are the factors to consider when you're thinking about how healthy your bumblebee populations are, though.
Thank you. The same listener also has a question. Does bringing in managed bees for pollination of orchards, market garden, or greenhouses potentially introduce pathogens to native bees?
That's a great question. The short answer is maybe / yes. So I guess what we need to consider is how pollinators that are managed are managed. And so it really depends on how well they're managed and how healthy they are. In many cases, managed bees live in higher densities that can promote pathogens, and some pathogens, not all pathogens, can be transmitted at the flower, which is to say if an infected pathogen or a colony of any type, whether it's bumblebee, honeybee, or solitary bee, does contain that pathogen, they can sometimes leave those pathogen cells at flowers where wild bees might interact with those flowers and connect and collect those pathogens. So it is possible. We do have some data to support that for bumblebees, commercial bumblebees, and this pathogen called Crithidia, where we do see higher levels of Crithidia, which is a gut pathogen, in native bees, wild bees, around greenhouses. So there is some evidence for that. but it would depend on the pathogen, and it's not necessarily going to happen. It's just it could happen.
Thank you. A listener named Ken is wondering how far into the canola fields were the collections made, and were you sampling the pollinator community well into the field, or possibly sampling a mix of bees exploiting the canola exclusively and those that were only transient in the canola but can be considered as non-canola preferring?
Good question. So canola, as you know, gets really dense really fast. And so my students were collecting pollinators on the very edge of the canola field, as well as 5 meters into the canola field. I think that's a really good point, that some of the pollinators we see in canola fields are using canola transiently, and some of them are more dedicated to canola. What we can tell you for sure is that pollinators near canola fields tend to focus on the edge of canola fields, and that's in part because they always need to get back to their nests, which won't be in the middle of the field. They'll be on the edge. So the data we have on pollinators near canola fields or in canola fields will be both that edge population and bees that are using this resource, but on the periphery. I hope that answers that question.
Yes, and our listener Sally would like to address the elephant in the room. If indeed the challenge to conserving the bee populations is rooted in habitat loss and pesticide use, how can these issues be addressed in an impactful way?
That's an excellent question. So habitat loss is definitely a huge part of conserving bees. The good news is what we find is bees don't need a lot of natural habitat in order to persevere. And so one of the major recommendations that is frequently made in sort of intensive agricultural areas is quite simply leaving strips of land that are not developed. This is not always an approach, this is an approach that still needs to be considered. It still requires work on the part of growers, but having that additional natural habitat can be a huge benefit to pollinator communities because it provides that important habitat and usually also provides a little bit more consistency across floral resources. Sometimes this natural habitat is as simple as leaving weeds in the ditch around fields instead of spraying them, and that can make a huge impact on whether or not pollinators are happy. Pesticides are definitely an elephant in the room. They're a big challenge. We certainly have more and more evidence suggesting that pesticides, even when sprayed away from flowering time, are problematic. And I would argue that I don't have a great solution to that at the moment. But it's definitely something that comes up repeatedly, and the good news is it's something that we're all thinking about. And so I think we'll keep working on it, for lack of more specific things to say, and I think it will be one of the challenges in trying to find a way for agriculture and conservation to come to the table together.
Great, thank you. One of the listeners is wondering if you could go back to the graph that you showed very early in the presentation, and the graph is showing the importance of other insects versus honeybees as pollinators.
Or this is too fast, you guys.
And they're just wondering if you could explain the vertical axis on that graph.
Sure. So this vertical axis, the beta or the y-axis is it's a regression coefficient is what it specifically is, but essentially it is a way to standardize the metrics across all these studies. So if we were trying to compare almond to buckwheat to coffee, those are crops that produce different numbers of seeds, different amounts of seeds. Some of the metrics in this graph are also, some of them are seed set, some of them are fruit set, some of them are number of visits. And so that regression coefficient you can just consider to be a standardized value. It's not dissimilar to an effect size, which is essentially where we are trying to standardize all of the information across some very disparate groups in order to compare their impact. So it tells us about impact, relative impact. I hope that clarified that a bit.
Yes, I think so. There's another listener wondering if you have studied the effect of restoration or reclamation in industrial disturbance, such as pipeline or power line, on bees' diversity.
So I have not. I do believe some groups are doing that. So there's definitely work, I don't know about in pipeline, sort of in northern prairie environments. There's definitely work on studying pollinator communities in highly disturbed areas. Those can include highly managed agricultural areas. There's work done on urban communities. I don't know off the top of my head if sort of pipeline and oil fields reclamation sites have been included in those studies. It would be an interesting thing to look at. Many of the sites that we would think of as reclamation sites in Alberta, I'll specifically speak to Alberta because I know it a little better, are not too far north for pollinators per se, but not necessarily the habitat that would be dominated by pollinators. And so sometimes those sites, like you can see even in our more boreal sites, we just see fewer pollinators in general. But it'd be a fascinating question, I think an important one as well.
Good, thank you. And our listener Courtney is wondering, for the study, are there sample methods that you're aware that could be conducted on live bees, such as using behavioral characteristics for classification? And she's also wondering why the sample sizes for the study is so large.
Aha. Those are excellent questions. So let me do the second question first. So believe it or not, those number of specimens is relatively small for the type of study that we're doing. Collaborators of mine have collected anywhere between 50 and 100,000 specimens within their sampling periods. And so essentially our goal is to collect everything we can for a short period of time. I think it sounds like a lot of bees, and it is, 19,000 bees, but if you divide that across 285 site visits, it becomes a lot less momentous. So in terms of the actual population of bees at a per site basis, where we're only monitoring bees twice a season, we're not actually taking that many individuals out of the population. But yes, it does seem like a lot of bees, doesn't it? Again, as I say, I have colleagues who have studied whether or not these type of surveys seem to impact populations of bees, and the evidence is no, but again, that's based on studies that last only a few years, not many, many years. As for the first question, whether or not we can identify bees that are live, it depends on the level of resolution that we want. So there are certain bees that are easy to identify. They're very easy to identify to genus in some cases, so I can always tell you what a bumblebee is compared to another bee. We did actually identify all of the honeybees in our study as live bees. They're very distinct from all the other bees, and so we did not capture any honeybees in our study whatsoever. But we did count them. I didn't show you that data, but we did count them. And that's in part because they're a managed species. They are, they are an animal that belongs to a grower, and we didn't want to harm them. In terms of identifying pollinators, particularly bees to species, that is very difficult to do without looking under the microscope. And so we are limited if we would like to look at the actual biodiversity of the community, and in particular for looking for certain groups. We are limited to surveying using these more these less friendly methods, these methods where we actually sacrifice our specimens.
Thank you, Jessamyn. One of our listeners, Dan, has several questions. So first of all, some years ago, he placed pan traps around western spiderwort and also did a lot of direct observations and found that although halictids were common pollinators, flies were very common, maybe more so. Does this seem likely to be important to pollination on the prairies? Several other non-bee groups were also in the flowers.
That's a great question. You know, we don't know a lot about fly pollination, and we know very little about fly pollination in the prairies. I think flies are very important pollinators of certain groups of plants, but that is a restricted group in general. I would also argue that flies are, we don't have a lot of data on whether or not flies are really good visitors or really good pollinators. So they may be really good at visiting plants without actually moving pollen. But that being said, to be honest, we don't know a lot about fly pollination in grasslands, to my knowledge. It's likely that bees are playing a pretty important role, but I don't know that I can speak to how important flies are at this point.
That's interesting. Thank you. Another question by Dan. He says, as someone with decades of experience in testing non-target effects of insecticides, including toxicology, he is always mystified that attention to the problem took so long, but also that sorry, my pronunciation here is bad, imidacloprid insecticides seem to be the only ones mentioned. Pyrethroids are highly toxic to bees and were on your slide but often not mentioned.
Yes, good point, Dan. So a couple of things. I think part of the reason that imidacloprid, which is one of these new neonicotinoids or neonics, gets more attention than sort of more traditional pyrethroids, there's a couple of reasons. One is that growers have moved very significant, my understanding is growers have moved very significantly towards neonics. Neonics has sort of become the new attractive pesticide. They're highly effective against pests and can be easily distributed to crops either through seed treatments or through spraying treatments. And so I think part of why they've gotten so much more attention is just how quickly they came online and how quickly they're being used across, not just across the prairies, across the world. Pyrethroids definitely do have effects on bees, but I think, yeah, I couldn't tell you exactly why they didn't get as much attention, except that neonics have become so prominent so quickly. They've risen so quickly amongst the ranks. We've known that neonics can be problematic for some time. One of the challenges with neonics has been that the data have been mixed, and part of why the data have been mixed, to my knowledge, is that data have been collected in natural habitats and in the lab. And to be honest, in the lab, neonics are not so bad. They don't kill everything outright, and they certainly don't kill pollinators at the concentrations that we see, at least consistently kill pollinators at the concentrations we see in fields. However, more recent data are suggesting that those lab studies have this very artificial environment where pollinators are only being challenged by pesticides, and they have all this other food and all these other good things happening. And then what we see is in natural environments where pollinators are under stress, that the effects of these pesticides are greatly increased. We're also starting to see a lot of sublethal effects that haven't previously been reported. So a lot of the time we just try to kill pollinators, and we use that as information, you know, okay, this chemical killed pollinators at this level. But a lot of new data is suggesting that the exposure to pesticides can affect learning and ability to collect floral resources, ability to support a colony, ability to fly, and those are harder to measure but equally important and can have pretty detrimental effects. So the good news is that this issue has come to the forefront, and it's becoming something we're all talking about.
Great, thank you. And the last question by a listener named Lewis is, he's just wondering if he can get a copy of some of the slides.
Can you get him to send me an email, and I will provide what I'm able to.
Okay, perfect. And I think that's it for the questions. So with that last one there, I'd just like to mention to all of our listeners that this presentation is being recorded, and it will be uploaded to PCAP's YouTube channel, and that's youtube.com/user/skpcap. That's also available on our website as well, or if you check the PCAP Facebook page, there'll also be links to this presentation. So I just want to thank you very, very much, Jessamyn, for the fascinating presentation today. And I'd also like to thank the audience for tuning in. And just to stay tuned for other events going on with Prairie's Got the Goods Week. So thank you all very much, and have a great rest of your day.
Thank you.
Thanks. Bye.